and extending lifespan that way. And really what you want to do is stabilize your epigenome or reboot it, reset it. And the analogy that I used in my book is this, the CD analogy, that the DNA is the music and aging is the scratches, so that the cells really can't read the right genes, or in this analogy, the songs are skipping around and cells lose their ability to function. We find that old brain cells from animals are more like skin cells. You know, of course you're not going to be able to think correctly if your brain forgets what it's supposed to do. The good news is that we think there's a way to polish off those scratches, or in the body, remind our old cells how to work again and be young again, and literally be young again. Senescence is another outcome, I think, of epigenetic noise. When cells lose their ability to function and reach a state where they are jeopardizing the organism, potentially becoming cancerous, there are mechanisms to shut those cells down. And this is what's called cellular senescence. And in this diagram, you can see on the right, these pink cells are accumulation of senescent cells. And so many of you will have heard of this, and there are ways, both with supplements and supplements plus drugs, that appear to be very effective in getting rid of these cells. And if you do that in a mouse, the mice are rejuvenated. Here are the clocks. We build these all the time in my lab. We do it for mice. We can do it for their blood, their skin. We've got a human clock. There's so many clocks, it's not even, it's even hard to keep up. And the clocks are getting better and better at predicting health and longevity, but also other things such as future metabolic health, future fertility. And this is what we're working on. The other thing we have is a breakthrough in reducing the cost of these tests. Right now, to run them, it probably would cost most people a few $100 commercially. At best, you'd be down to $100 per person. We can now multiplex. We can pool 1,000 people, run it for the same cost as one. So this test is going to be essentially free in a couple of years, which means that we can regularly test ourselves for what is happening when we take a supplement, when we exercise, when we change diet. And we're finding actually that these clocks first of all, they have to be very accurate, but when you get accuracy, then you also can see that they change. And this study from yesterday was a 24-month study, though I think we can probably see changes earlier. But within those 2 years, those who are on the healthy diet, quote unquote healthy diet, which, by the way, I wrote what was in the diet if you want to see on that tweet. So it's a lot of olive oil, the usual stuff. Don't overdrink. You can drink a bit of red wine. There was whole grains, cut back on red meat, don't eat processed meat, that kind of stuff. Those women had a much lower rate of aging than those that didn't adopt that diet. So this was a key experiment that we did. We had proposed that DNA breaks that break a chromosome distract the sirtuin epigenetic regulators, and they go to the break, and then they have to find their way back, and they don't do that efficiently. And we proposed that in a Cell paper in 2009, but then we had to test if that's true. And the hypothesis predicted that if we create chromosomal breaks that have to be put back together multiple times, even if we reestablish the genome, so there's no change to the DNA content, the epigenome will change, and it will cause aging. That was the prediction. And I remember people in the lab saying, these mice are going to die, they're going to get cancer, come on, that's ridiculous. But we like to do the ridiculous in my lab. The other thing that we like to say is we make the impossible possible. So we did the experiment. Actually, my job, the hardest part of my job is convincing people to do an experiment because they take a while. This is what's called an ice mouse. This is the control mouse. Its sibling was treated at age 5 months. A mouse typically lives a bit over 2 years, and then it was another 10 months after that treatment. We treat them for 3 weeks, and we accelerate their epigenome so that by 16 months of age, the treated mice look like this. And this is not just an old-looking mouse by every measure, histology, biochemistry, gene expression, and the biological clock. This mouse is older, and we've done thousands of them. There's It's highly reproducible. And now we're working on, well, we've slowed this down. We have ways of doing that, I'll tell you, but we also are working on reversing this process. One of the ways to reverse aging is to use a molecule that raises NAD levels. We showed early 2000s that in yeast, if you raise NAD production, the yeast cells live longer by turning on the sirtuins. The sirtuin enzymes need NAD. We need it for life. Sirtuins also need it. And they stabilize the epigenome, they repair DNA, and they protect mitochondria and proteins and telomeres. Think of sirtuins as the guardians of our body. But as we get older, we lose NAD production. We have less of it. In our skin, it's estimated we have half the levels at age 50 that we had when we were 20. You can raise the levels in an animal and in a human 2 to 3-fold by giving precursors to NAD. NAD itself, we can talk about how effective that might be, but these mice have been fed NMN for just 4 weeks in their water supply, and I think you can tell the one that was drinking NMN water versus the one that wasn't. The mice on the left could run 50% further, and when we gave it to young mice and exercised them, they ran so far the treadmill stopped working, and we thought we broke it. Turns out the software was never written for a mouse to run more than 3 kilometers, so we had to rewrite the software. So we figured out how, we published this in 2018, we figured out how it works, we think. The lining of the blood vessels gets old, it doesn't respond to muscle signals. And what happens to us, we know this, is that even if we exercise when we're old, we don't grow a lot of blood vessels, and our organs get starved for oxygen, particularly our brain and our muscles, and heart, which use a lot of oxygen. But when you give mice NMN, NAD levels go up. SIRT1, the equivalent of that yeast SIR gene, now allows the muscle and the brain to signal to the blood vessels to grow. So these mice on the left have more blood vessels like a young mouse, actually without even having trained.